Hi, we're Lab Group 3, and for our final video, we're going to show you how to use a micropipetter.
I'm Maggie, and I'm going to show you a demonstration.
I'm Vince, and I'm going to show you the parts of a micropipetter.
I'm Carrie, and I'm going to show you what not to do with a micropipette.
So, in Miss Anselmina's lab, you're going to be using one of 4 different micropipetters. This is the BioRad micropipetter, and it measures 1,000 microliters. And then you have the more expensive, more accurate Eppendorf micropipetters. You break it, you buy it. This one's 10 microliters. The yellow tip one is 100 microliters, and the blue is 1,000 microliters. So the parts of both of them are basically the same. This is, we call it the plunger, and you're going to use this to add your sample. This is the handle. You hold a micropipetter like this, and this button here is what you're going to use to eject the tip of the micropipetter. When you're ready to use it, all you do is you're going to take a tip and just kind of set the tip of the micropipetter right in like this, and you're going to get your tip, and then you're going to take your sample, and Maggie's going to show you how to do that. And then whenever you're finished, you just push this button and eject the tip. Same basically with the Eppendorfs, except they're special. So each Eppendorf is going to have its own tip. For the 1,000, there's 1,000 microliter tips, and same for the 100 and the P10. So that's basically the parts of a micropipetter, and Maggie's going to show you a demo.
I'm Maggie, and I'm going to show you a demonstration. First, I'm going to start with the BioRad, and the first thing you have to do is get a tip, which are all located in boxes and labeled boxes. And you just have to find the one for the BioRad. And to get a tip, you just need to stick the micropipette in, push a little bit, and pull out. The micropipette takes different measurements of microliters. And to change them, you can either use the top or right down here, the black knob. This one is currently at 670. And to take a sample, get down at eye level. And there are 2 stops when you push down on the micropipette. There's the first one, and then if you push all the way down is what you use when you release the sample. So you can only go to the first one. Push down to the first one, and then put the tip right above the break of the water, and just let the tip come back up. Make sure there are no air bubbles. If there are air bubbles, release the sample and take it again. To release it, put the tip into the cuvette and then push it up against the side and push all the way down past the first stop to the bottom. And then when you eject the tip, Vince showed you how you just press the button and put it into a beaker. For the Eppendorf, make sure you're using the correct tip for whichever one you're using, and those are all labeled too. It's the same idea. You can't go down the whole way. There's the first stop, and then if you push down the whole way, it releases the sample. And then put the tip into the cuvette up against the side and release, except that chocolate's staying up top. And then same idea to release the tip. Each micropipette in the Eppendorf are numbered differently, and depending on where the line is how much you have. This one's 4.7, this one's 470, and this one's 47.
I'm Carrie, and I'm going to show you what not to do with micropipettes. Whenever you dispense liquid, sometimes you'll have little air bubbles or droplets of water inside. Never lay it down on its side or hold it upside down because you can damage the micropipette and then you have to buy it yourself. Never play with lab equipment because you can always injure yourself or the micropipette. Do not leave stuff all over your lab table as you can injure yourself and tips can get contaminated. During labs, you do not want to use bad grammar, swear, or violate the dress code because Ms. Sansmino will yell at you. Whenever you're doing labs, you have to have proper shoes. Don't wear open-toed shoes. And if you have long sleeves, make sure you roll them up and pull your hair back.
So now we're going to run a demo for the lab that you're actually going to do with the micropipettes. First thing you want to do is obtain one 1.5 milliliter microcentrifuge tube, and you want to mass it using the analytical balance. So just slide back the door, put your sample in, and make sure you zero it with the empty microcentrifuge tube in there. Then what you want to do is you're going to pour about 15 milliliters of distilled water into a small beaker, which Maggie has right here. So now you're going to use the appropriate micropipette, and Ms. Sansmino will give you different measurements that you have to take. Transfer the first amount of water into the centrifuge tube.
Just a reminder, when you use the Eppendorf, the line on the reading acts as a decimal point.
And be careful to watch out for bubbles while you're taking this reading. So now once you've filled the microcentrifuge tube, put the sample into the analytical balance, and you're going to get a measurement for how much, the mass of how much water is in the tube.
What want me to do with the Biorad?
We're going to do one more, and we're going to use the Biorad, and we're going to show you the same process one more time again. Massing out the empty tube. Now Maggie's calibrating the measurement. Maggie's very precise with the measurements. And then once you obtain this data, you can do whatever necessary calculations you have to do. That Ms. Sansmino will give you. So fill the tube. Put it in. And again, that's going to give you the mass of the water that's inside the tube. And you can use that to determine how accurate you were with the micropipette.